Okay, so I guess when I go to share screen, it's going to close the whiteboard and then I'll have to open it back up. Okay. All right, so Gregor Mendel is considered to be the father of modern day genetics. Most of Mendelian genetics is based on a one protein, one gene model. A lot of human traits are going to be more complex than this. They're going to be based on multiple genes and multiple locations on chromosomes. But in order to understand the basics, we're going to start with Mendelian genetics, and then we'll get more complicated from there. So Mendelian genetics are considered to be genetically simple traits that are determined by alleles at a single locus. So alleles are going to be alternative forms of a gene. So genes will either be dominant or recessive when we're looking at the simple Mendelian traits. And the dominant alleles are going to mask the expression of the recessive. And we'll talk about the different genotypes that are possible. But essentially, the gene is going to be located at a specific area on the chromosomes, on the homologous chromosomes. So you inherit one chromosome from your mother and one chromosome from your father. And remember, as humans, we have 46 individual chromosomes or 23 pairs. So those pairs at each location, at location 1, 2, 3, 4, so on and so forth, those are the homologous chromosomes. So you inherit one of those from your mother and one of those from your father. Mendel has two laws: the principle of segregation that states that alleles will separate from each other during the formation of gametes. So when we learned about meiosis yesterday, or not yesterday, on Tuesday, when we learned about meiosis, we learned that gametes will separate from each other during the process of independent assortment and crossing over. That's essentially the reshuffling of the genes. So humans have the ability to create over 1 million different unique gametes. So egg cells and sperm cells. So this principle of independent assortment and crossing over will essentially allow for the resortment and shuffling of those genes. So the principle of segregation states that alleles will separate from each other during the formation of gametes. And the principle of independent assortment states that the presence of alleles of one trait will not affect the expression of another trait. So we're going to be looking at one trait at a time. So whether or not you have attached or free hanging earlobes won't necessarily affect whether or not you have a widow's peak or a straight hairline. And whether or not you can taste PTC won't necessarily affect whether or not you can roll your tongue. So we're just looking at one characteristic or one trait at a time. All right, so a gene is the segment of a chromosome's DNA that is coding for a specific protein. So when we learned about protein synthesis on Tuesday, transcription and translation, that process is coding for all of the alleles and genes that make up our genetic code. So the gene is the segment of a chromosome's DNA that's coding for a protein. An allele would be alternative forms of a specific gene. So there may be genes that code for light-colored blue eyes and other genes that code for dark-colored brown eyes. And then, of course, genes for everything in between. So the term homozygous is important. Homozygous means chemically identical forms of a gene. So when you break that word down, the first part of the word means same, and zygous means zygote. So thinking back to what we learned on Tuesday, a zygote is simply a fertilized egg. So when that sperm cell fertilizes the egg cell, you get a zygote. So that's where the 23 chromosomes from dad comes from and the 23 chromosomes from mom. So if at that location on the chromosome, you have chemically identical forms of the gene, and they're both dominant, then you would call that genotype homozygous dominant. So it means that the genes are the same and they're both dominant. So when we're using the basic Mendelian Punnett squares, the dominant genes will always be represented by uppercase letters. So homozygous dominant genotype would be represented by 2 uppercase A's, 2 uppercase E's, 2 uppercase W's, 2 uppercase R's. You guys get the point. Whatever trait we're talking about, 2 uppercase letters will always represent homozygous dominant. And then homozygous recessive will always be represented by 2 lowercase letters. So in this example, we're looking at 2 lowercase a's, but it could be 2 lowercase e's, 2 lowercase w's, 2 lowercase r's. whatever trait we're looking at, 2 lowercase letters will always represent homozygous recessive. All right, the only other genotype that we will look at least for the basic monohybrid Mendelian traits, will be heterozygous. So break that word up again. Hetero means different, and zygous means zygote. So again, zygote is a fertilized egg. So that means that you inherited a dominant allele from one parent, either mom or dad, and then you inherited a recessive allele from the other parent. So heterozygous simply means chemically alternative or different forms of a gene. And in this case, the dominant allele will actually mask the recessive allele in the phenotype. So the phenotype is going to be the physical expression. So in this example here of the 2 babies, their phenotype, we're looking at eye color. So their phenotype is simply whether or not they have the allele for blue eyes or they have the allele for brown eyes. Okay, so when we're looking at each trait, the phenotype is simply going to be the physical expression for that trait. So it might be whether or not your earlobes are free hanging or attached. It might be whether or not you have a straight hairline or a widow's peak, whether you have a cleft chin or no cleft chin, whether you have a hitchhiker's thumb or a straight thumb. You know, that's what we mean by phenotype. It's the physical expression for just that trait. So we're looking at just one physical characteristic or attribute at a time. Okay, so this term genotype, think genetic. So genotype is the genetic makeup of the individual. So with these basic traits that we're looking at for the monohybrid crosses, there's only 3 possibilities. The genotype will either be homozygous dominant, represented by 2 uppercase letters, heterozygous, represented by 1 uppercase and 1 lowercase, or homozygous recessive, represented by 2 lowercase letters. Those are your only 3 possibilities for these simple monohybrid crosses. And like I mentioned, the phenotype will be the physical expression of that genotype. So an example would be attached earlobes versus unattached, straight hairline versus widow's peak, blue eyes versus brown eyes, straight thumb versus hitchhiker's thumb. You guys get the picture. So when we're doing these Punnett squares, we're always going to match up the genotypic ratio in a certain way. So the genotypic ratio will always be the number of homozygous dominants versus the number of heterozygotes versus the number of homozygous recessives. So in this particular example here, let's go to back to our whiteboard. So we're going to go here to stop share, and hopefully that whiteboard will come back up. Oh, no, it's going to make me do it again. Oh, wait, no, here it is. Okay. All right. Are you guys able to see the whiteboard again?
Yeah, we can see it.
Yep.
Okay, perfect. Okay, so when you're doing a Punnett square, you're going to put one parent, doesn't matter whether it's mom or dad, you're going to put one parent along the top and another parent along the side like this. Doesn't matter whether mom or dad is on top, doesn't matter. You just need one parent along the top, one parent along the side. So if we have 2 parents that are heterozygous for earlobes, and for earlobes, let me write out the possible genotypes and phenotypes for you. So for humans, you could be homozygous dominant, so 2 uppercase E's. You could be heterozygous, so 1 uppercase, 1 lowercase, or you could be homozygous recessive, 2 lowercase letters. Okay, now the phenotypes, there's only 2 possible phenotypes. You're either going to have free hanging earlobes. So if you have free hanging earlobes, that's the dominant. It's the dominant phenotype. So if you have the dominant phenotype, you could either be homozygous dominant or you could also be heterozygous. Those are your 2 possibilities. Okay. And then for the Punnett square, so we're simply going to be doing the parents' genotypes along the top and the side. So in practice problem number 1, we have 2 heterozygous parents. Both mom and dad are heterozygous for earlobes. So that means along the top and along the side, we need to represent mom and dad's genotype with 1 uppercase letter and 1 lowercase, just like that. 1 uppercase, 1 lowercase. So each little quadrant, each little section of this Punnett square, see how it's basically a rectangle divided into quadrants. So each little section is going to represent 25% of generation 1. Okay, so each little square is representing 25% of generation 1. And bear with me, guys. This is my first time using the whiteboard, so bear with me. Not in real life, but on Zoom. This is kind of cool, right? We can use the whiteboard in Zoom as well. So basically, each little quadrant represents 25% of generation one. So we're basically calculating the likelihood that that parents would produce various genotypes. So we're calculating the likelihood that they will have children with attached or unattached earlobes. So here in this first little box, we're going to take one from here and one from here. So in this first little box, we have a homozygous dominant possibility. Okay, so we took one letter from here, one letter from here. And you're always going to write the uppercase letter first, but in this case, we have two uppercase letters, easy peasy. So we had two uppercase letters in that first box. In the second box, we're going to take one from here, one from here. So we have one uppercase and one lowercase. So that's a heterozygous child there. And then here in this box, we're going to take one from here and one from here. So we've got another heterozygous possibility. And then in this last box here, we're going to take one from here and one from here. So we have a homozygous recessive possibility here. Okay, so now we have our Punnett square. So that's giving us the likelihood of their children, essentially, the probability that they would have a child that's homozygous dominant versus heterozygous versus homozygous recessive. And then what we can do next is then do our genotypic ratio. So our genotypic ratio will always be set up in the same way. It's going to be homozygous dominant versus heterozygous versus homozygous recessive, like that. Okay, so once you have it set up like that, all you have to do is count. So out of those four little boxes, how many homozygous dominants do we have? Just one, right? I'll do the first one for you. We just have just one homozygous dominant there, right there. Okay, just one homozygous dominant. And then, ooh, all right, so we have one homozygous dominant. How many heterozygotes do we have? We have 1, 2 heterozygotes, so we're going to write a 2. And then how many homozygous recessives do we have? We have just one. So our genotypic ratio is 1 to 2 to 1. Now you can also write that in percentages. If percentages make more sense to you, you can also calculate it in percentages. And let me show you how to do that. Okay, so it can be 1 to 2 to 1. But what's 1 divided by 4? Because there's 4 little boxes, so 1 out of 4 would be 25% of generation 1. 2 out of 4 would be 50% of generation 1. And 1 out of 4 again would be 25% of generation 1. So essentially what that means, every single time this mom and this dad produce a child, they have a 25% chance of having a child that is homozygous dominant for earlobes. They have a 50% chance of having a child that is heterozygous for earlobes. And they have a 25% chance of having a child that's homozygous recessive or attached earlobes. So that's the genotypic ratio there. Okay, now phenotypic is going to be just slightly different. Oh. Because remember, a phenotypic ratio is talking about the phenotype. So in this case, we're simply looking at whether or not that child has free hanging, which is the dominant, free hanging versus attached earlobes. Okay, so there's only 2 possibilities for the phenotype. So now remember, when we're talking about the basic monohybrid crosses, as long as there's at least one dominant, uppercase E, that means that that child will have the dominant phenotype, which is free hanging earlobes. So out of those 4 little boxes, there's at least 3 of those children, 3 of those boxes have one uppercase E. So that means that 3 of those children will have free hanging earlobes. Okay, and only 1 of those children would have attached. Okay, so the phenotypic ratio is 3 to 1, or if you want to write it in percentages, you can write it as 75% versus 25%, because 3 divided by 4, 75%, 1 divided by 4, 25%. So essentially what that means, every single time this mom and this dad produce a child, they have a 75% chance of producing a child that has free hanging earlobes, and they only have a 25% chance of having a child with attached earlobes. Okay, is that making sense? You some questions so far? Just maybe give me some thumbs up if this is making sense. Perfect. Okay, so let's do the next one. Okay, so practice problem number 2 is about tongue rolling. So in practice problem number 2, we're going to be calculating the likelihood of generation 1 as far as tongue rolling goes. So in humans, the ability to roll your tongue is actually influenced. It's also environmentally influenced, we're learning, but it is influenced by a dominant gene. So being able to roll your tongue would be the dominant form over being unable to roll your tongue. Okay. And sorry guys, I set these up all ahead of time, and then of course, looks like they shifted somehow. But that's okay, we can draw them here on the whiteboard. Okay, so tongue rolling is dominant, and the inability to roll your tongue will be recessive. All right, so in this scenario, we're crossing one parent that is heterozygous. So remember our vocab words. Heterozygous means that there's one dominant allele, so that's represented by an uppercase R, and one recessive allele, represented by a lowercase r. So we've got the heterozygous parent up here at the top, and then the other parent is unable to roll their tongue. So the only way they would be unable to roll their tongue, they would have to be homozygous recessive. Because remember, in humans, tongue rolling is dominant. So if you're a tongue roller, that means that your genotype could either be homozygous dominant or you could be heterozygous. So the only way you're a non-tongue roller, that's the recessive trait. So we'll just put non-roller here. So if you're a non-tongue roller, then you have to be homozygous recessive. You have to have 2 lowercase letters there. Okay, so in this case, we're crossing a parent that is heterozygous with a parent that is a non-tongue roller. Okay, and this is practice problem number 2 from check-in assignment number 1. Okay. All right, so here in this first little box, so now we have it set up. We've got one parent along the top and we've got one parent along the side. Here in this first little box, we're going to take one from the top and one from the side, always putting the capital letter first. We've got one big R and one little r. Okay, so that's our first little box representing 25% of generation 1. In our second little box, we take one from here, one from here. So we've got 2 little r's, a homozygous recessive child in that second box. And then here in this box, we're going to take one from here and one from here. So we've got another heterozygous child in this little box. And then in the last one, we're taking one from here, one from here. So we've got a homozygous recessive in this little box here. Okay. All right, so that's our Punnett square. So remember genotypic ratio, thinking back to the vocab words we just learned in the PowerPoint slides. So we've got the genotypic ratio is always set up like this: homozygous dominant versus heterozygous versus homozygous recessive. Always, always, always set up like that. So out of those 4 little boxes, you guys tell me, do we have any homozygous dominants?
No.
Nope. So what we're going to write is we're going to write a 0. Okay, so no homozygous dominant, so we write a 0. Out of those 4 little boxes, you guys tell me, are there any heterozygotes?
Yes.
Yep. 50%. How many? 50%, or we can write a 2. Are there any homozygous recessives?
2.
2, beautiful. So it can be, you can write it as 0 to 2 to 2, or if percentages make more sense to you, you can write 0%, 50%, and then another 50%. So essentially that means every single time this mom and this dad produce a child, they have an equal likelihood of having a child that can roll his or her tongue or not roll his or her tongue. So it'd be pretty even odds. If you were placing bets, it'd be a 50-50 bet whether or not this parent and these two parents produce a tongue roller or non-tongue roller. Okay, so the genotypic ratio is 0 to 2 to 2, or you can write it as 0 to 50 to 50. Either way, whatever makes more sense to you. Okay, now if you're doing phenotypic ratio, so phenotypic is talking about the phenotype. So we're just simply going to write tongue roller versus non-tongue roller, because that's what we're looking at here. We're just looking at whether or not they can roll their tongue or not roll their tongue. Okay, so tongue rolling is the dominant. So that means as long as there's at least one dominant uppercase R, that child will be able to roll his or her tongue. So out of generation 1, out of those 4 little boxes, how many tongue rollers would we have?
2.
2. And how many non-tongue rollers would we have?
2.
2. Or if we want to write it as percentages, easy peasy, piece of cake. What percentage? What's 2 divided by 4?
50.
50%. That's just showing there's an even likelihood, equal likelihood of a tongue roller versus a non-tongue roller for this particular Punnett square. All right, you guys with me? Is this making sense?
Yes.
Yes, okay. All right, so let's do the last one. The last one of the monohybrids, and we're gonna make it a little bit more complicated. Let's do one more with the monohybrids. So this is practice problem number 3 on check-in assignment number 1. Okay, so this one is looking at whether or not you can taste PTC. So PTC is a chemical that allows you to taste bitterness. So PTC tasting, so tasting is dominant. So if you're a taster, that means that your genotype is either homozygous dominant, we're going to use T's this time, or you could also be heterozygous. And then if you're a non-taster, that's the recessive condition. If you're a non-taster, the only possibility of being a non-taster, you'd have to be homozygous recessive. So you would need 2 lowercase t's. Okay, so those are your 2 possibilities. And in this Punnett square, we're doing a parent. One parent is homozygous dominant. So if they're homozygous dominant, they need 2 uppercase t's like that. Oops, no. Or the one parent is homozygous dominant and the other parent is a non-taster. So if they're a non-taster, they have to have lowercase t's like that. Okay, so we're setting up our Punnett square. One parent is along the top, one parent's along the side. Doesn't matter whether, you can put mom along the top or along the side. Just you need one parent along the top, one along the side. Each little box is representing 25% of generation 1. So here in this first little box, we're going to take one from here, one from here. So what would go there? A big T and a little t. And then in the next box, we take one from here, one from here. So what goes in this box here? Big T, little t. And then in our third box here, we take one from here, one from here. What goes in this third box? Big T, little t. And then in our fourth box, we're going to take one from here and one from here. So in our last box, we've got a big T and a little t. All right, so this particular cross results in 100%. All of their children are going to be heterozygous for tasting, for PTC tasting. But we still want to set up our genotypic ratio in the same way. We still want to set it up as homozygous dominant versus heterozygous versus homozygous recessive. Okay. All right. So out of those 4 little boxes, you guys tell me, how many homozygous dominants do we have?
None.
There's 0.
0. So we write a 0. Out of those 4 little boxes, how many heterozygotes do we have?
4.
4, so we write a 4. Out of those 4 little boxes, how many homozygous recessives do we have?
0.
0, none.
Beautiful. Nice job, guys. Excellent work. All right. Now, if you want to do percentages, easy peasy, right? 0 divided by 4, 0%. 4 divided by 4, 100%. 0 divided by 4, 0%. So essentially what we've determined every single time this mom and this dad produce a child, 100% of those children will be heterozygotes. 100%. There's not a chance of them having any other genotype. Okay, so that's our genotypic ratio. Phenotypic is the phenotype. So in this case, we're simply looking at tasting versus non-tasting. We're going to do tasters versus non-tasters. So remember, tasting is dominant. So as long as there's at least one uppercase big T dominant allele, then that child will be a taster. So you guys tell me, out of those 4 little boxes, how many taster children would we have?
4.
4. There's at least one big T for all of those children. So all of them will be tasters. None of them will be non-tasters. Or if you want to write as a percentage, 4 divided by 4, 100%. So 100% tasters. And then 0% non-tasters. All right, you guys with me so far? You ready to make it a little bit more complicated?
Yeah.
Yes. Okay.
Uh,
All right, let's make it a little bit more complicated. All right, let's go back to our Punnett square here for a second. Or back to our PowerPoint, excuse me. Okay, so now we're going to talk about ABO blood typing. So ABO, every single human has an ABO blood type and an Rh positive, Rh negative. So to avoid confusion, we're going to talk first about the ABO blood type. We will get to the positive and negative, but for now, we're going to talk about ABO blood typing because ABO blood typing is determined at a separate location on the chromosome versus Rh negative, Rh positive. So this is also an example of codominance in humans, and I'll tell you why. But every single person has one of 4 blood types. You will either be blood type A, blood type B, blood type AB, or blood type O. And the combination of your alleles that you've inherited from your parents will determine your ABO blood type. So remember, on the homologous chromosomes, we're inheriting one of those from mom and one of those from our father. So each person will inherit either an A, B, or O allele from their mother, and each person will inherit either an A, B, or O allele from their father. And the combination will determine that person's blood type. So there's actually 6 possible genotypes here, but only 4 possible phenotypes. So the phenotypes, that's your blood type here. That's your phenotype. And the genotype is the letters. So if you have blood type A, you actually have 2 possible genotypes. You could be homozygous dominant. You could have 2 dominant A alleles, one from mom, one from dad. Or your second possibility, you could inherit an A allele from one parent and a B allele from the other parent. So in that scenario, that A allele is going to be dominant over the O. So either way, either of these combinations ultimately result in blood type A. Same deal for blood type B. You can inherit a B allele from both mom and dad, or you could inherit a B dominant allele from one parent and an O recessive allele from the other. But either way, whether you're BB or BO, that will ultimately result in blood type B. The codominant condition would be if you inherit an A allele from one parent and a B allele from the other. So that's called codominant because both of those alleles will be expressed in the phenotype. AB genotype results in AB phenotype. And the only way you end up with O, which is the recessive gene, so the only way you end up with O blood is if you have a copy from both mom and dad. So you would need an O allele from both mom and dad in order to have type O blood. Okay, so let's go back here to our whiteboard. Stop share. And then whiteboard. Okay, let's scoot this down for a second. Make you guys a little chart. Actually, we could do it here. Okay, so I'm going to write phenotype on one side here. And then I'm going to write genotype here. Okay, so the possible phenotypes. So we're just talking about the blood types. You could be blood type A. You could be blood type B. You could be blood type AB. Or you could be blood type O. Okay, so ooh, what happened there? Oh my goodness. Okay. All right, so if you're blood type A, your two possible genotypes, you could be AA, or you could also be AO. Okay, so those are your two possible genotypes if you're blood type A. If you're blood type B, you also have two possible genotypes. You could be BB, like that, or you could be BO. If you're blood type AB, you only have one possibility. Your genotype and phenotype will be the same. So if you're phenotype AB, your genotype is also AB. And then if you're blood type O, the only possibility that you would be blood type O, you would have to have two copies of the recessive allele. You would need to be OO. Oops. All right, you guys with me on the possibilities?
Yes.
Yes, okay. All right, so once we understand the possibilities, then we can also use a Punnett square to determine the likelihood of having children with the various genotypes and phenotypes. Okay, so in the first problem, we have one parent that is blood type AB. So we're going to represent that with an A and a B. So we're putting one parent along the top. And then we have another parent that's blood type O. So the only way that they can be blood type O, that means they need two copies of the recessive allele. They need an O and then they need another O. Okay? Then once you have it set up, it's going to be the same scenario or the same procedure as the other Punnett squares, putting the dominant allele first. So we're going to take one from here and one from here. So in that first little box, we have an A and an O. In that second little box, we're going to take one from here, one from here. So in the second box, we have a B and an O. In that third box, we're going to take one from here, one from here. So in that third box, we have an A and an O. And in our last box, we're going to take one from here, one from here. In our last box, we have a B and an O. Okay, now on these, you don't have to do genotypic and phenotypic ratios. We're just going to do percentages. So out of generation number one, we're just going to determine the likelihood of each particular blood type. So out of generation one, let's start with blood type A. How many of those children would have blood type A?
50%.
50%, beautiful. So out of generation one, 50% would have blood type A. All right, now out of generation one, what percentage of the children would have blood type B?
50%.
50%, perfect. All right, would any of those children have blood type AB?
No.
Nope. So blood type AB would be 0%. Out of generation one, would any of those children have blood type O?
No.
So 0% for blood type O. The type O equals 0%. Okay, so that's all you need to write for the blood type A, B, and O. You don't need to do genotypic and phenotypic ratio. You're just giving me the percentages for each potential blood type. Okay, so 50% type A, 50% type B, 0% AB, and 0% O. So essentially what that means to us is every single time this mom and this dad produce a child, they have an equal likelihood of having a child with blood type B or blood type A. Okay, equal chance. All right, now with some of the ABO blood typing though, we're gonna need to do more than one Punnett square in order to come up with our answer. And I'll give you an example here in just a moment. So the next one, so now we're on practice problem 2 underneath the ABO blood typing. So the practice problem is telling you that one parent has blood type A, the other parent has blood type B, and that they have a child with blood type O. Is that correct? Okay, so in this particular example, if one child or if one parent has blood type A, the other parent has blood type B, but we know for a fact that the child has blood type O. So we can actually do 4 Punnett squares to make sure we're looking at all possibilities. So of course, if one parent has A and one parent has B, it could be an AA crossed with a BB, like this. Okay, so in the first little box, if it's this possibility, that means, what would we put here in the first box? An A and a B, right? So that's a child with type AB blood. What will we put here in this box? Another A and another B. So another child with AB blood. And then here in this box, we've got an A and a B. And then here in this box, we've got an A and a B. Okay, so if it's this scenario, if one parent is AA, the other is BB, 100% of their children would be type AB blood. So this can't be possible, right? Because we know for certain that their child, they have a biological child that is type O. So this cannot be mom and dad's genotype. So to go back to the drawing board. Okay, let's say that maybe dad is BO instead of BB. So let's put an O there. Okay, so in this scenario, let's do our Punnett square again. What would go here in this first box? An A and a B. What would go in the second box? An A and a B. What would go in the third box? An A and an O. And what would go in the last box?
I know.
An A and an O. Beautiful. Okay, so in this particular scenario, 50% of the children would be type AB, and the other 50% would be type A. But this still can't be our possibility because we know for a fact that these two parents have a child with type O blood that's a biological child. So we know this cannot possibly be our parents' genotype. So we have to go back to the drawing board. Okay, so what if, let's go back to dad having BB. But let's say that mom has AO. Okay, so let's go back to the drawing board and say it's mom that's AO. All right, so we do our Punnett square. What would go here in the first box? An A and a B. What would go here in the second box?
Uh-oh.
A B and an O. What would go here in this box?
AB.
An A and a B. Oops. And what will go in our fourth box?
BO.
BO. So this still isn't our answer because we have 50% of our children, 50%, 2 out of 4 would be blood type AB, and 50% of the children would be blood type B. Okay, because as long as there's at least one uppercase B or A, even if there's an O there, that uppercase dominant B or A is still going to mask the expression of the O. Okay, so there's one more thing we can try, one more genotype. So let's say that mom is AO, but let's say that dad, instead of being BB, let's say that he is BO. All right, so let's do our Punnett square one more time and see if that's the answer. So what goes here in the first box?
AB.
AB, beautiful. What goes here in the second box?
AO.
BO. What goes here in this box?
AO.
AO. And what would go in our very last box?
OO.
OO. Perfect. So there's our answer. So these parents, if they are AO and BO, they actually have an equal likelihood of all 4 genotypes, or all 4 blood types, I should say. They have a 25% chance of having a child with AB blood. They have a 25% chance of having a child with BO blood. They have a 25% chance of having a child with AO. And they have that 25% chance of having a child with blood type O. Okay. All right. So that is actually my parents' scenario. My mom has blood type A and my dad has blood type O, or blood type B, excuse me, but I have O and my sister has AB. You guys with me so far? All right, one more with the ABO blood typing, and then we're going to move on to the sex-linked. And the sex-linked are a little bit complicated. They're not hard, but it does definitely take a little bit of practice. So the last one on the ABO blood groups. So we're looking at a dad, I believe it's dad, or it's essentially just one parent that's blood type B. And then there's another parent, I believe it's mom, that's blood type O. And it wants you to do the percentages for both. So we know so for the parent that's blood type B, there's actually 2 possibilities for that parent, right? That parent could either be BB, like that, or, oops. All right, guys, the whiteboard is going all over the place. Let's just draw it in. Okay. All right, so we're going to draw our Punnett square here. This. All right, so let's do our first possibility. We could have a parent that's BB, and then the other parent is O. So the parent that's O, their only possibility, they would have to be O and O. That's the only way that they would end up with type O blood. They need both copies of the recessive allele. So in this scenario, what would go here in this first box?
BO.
BO, beautiful. What would go in the second box?
BO.
BO, beautiful. What would go in the third box?
BO as well.
BO as well. And what goes in the last box?
BO again for the last time.
BO again, beautiful. Nice job, guys. So for this particular scenario, if one parent is BB and the other is BO, 100% of their children would be blood type A. Okay, or blood type B, excuse me. So 100% would be blood type B. Okay, it would be 0% for the other 3. So type B is 100%, type A would be 0%, type AB would be 0%, and type O would be 0%. Okay, but there's actually a second possibility for this particular scenario. The parent that's type O, we can leave that one alone because they have to be OO if they're O. But if they're B, they could be BB, or their second possibility, they could also be BO. So let's try it with that scenario. So let's say the type B parent is one capital B and then an O. So in this scenario, what would go here in the first box? A B and an O. We'll go here in the second box. An O and an O. And here in this third box, we've got a B and an O. And then our last box, we've got an OO. So for this scenario, if one parent is BO and the other is OO, then we have an equal chance. We have type B would be 50%. And we also have a 50% chance of having a child that's type O. Okay, so in this scenario, these two parents have an equal likelihood of having a child with blood type B versus a child with blood type O. Equal chance. All right, how you guys doing so far?
Good.
Good. Does anybody know their parents' blood type and want me to do their Punnett square for them? Or have another scenario you want me to do? Okay, let's move on to sex link just to make sure we have time for it. We're going to go back here to share screen. And we're going to go to the sex linked Punnett squares. So these ones are probably the most difficult. They're not, but they're still not crazy difficult. So let's talk about the sex linked ones to make sure we have time. So all the ones we're going to do, these ones are probably the toughest, just because you have to be careful about what the question is asking. Okay, so first let's talk a little bit about what sex-linked traits are. So the term sex-linked just means that the gene and the alleles are located on one of the sex chromosomes. So thinking back to what we learned on Tuesday, we have 46 pairs of 46 chromosomes and 23 pairs. So pair number 1 through 22, those are called the autosomes. The 23rd pair is the X chromosomes, or the sex chromosomes. So you're either XX if you're biological female, or you're XY if you're biological male. Now there are some aberrations, some differential options for that. We're not all XX and XY. There's actually multiple possibilities. So even sex is not purely biological. But just to avoid confusion, let's focus in on the typical condition first. Okay. So sex-linked simply means that the gene is located on one of the sex chromosomes. So sex-linked traits can either be X-linked, meaning that the gene is located on the X chromosome, or they can be Y-linked, meaning that the gene is on the Y chromosome. Most sex-linked traits are X-linked simply because the X chromosome is much larger and contains thousands of genes, whereas the Y chromosome only contains about 100. Most of the genes located on the X chromosome actually have very little to do with female sexual development and are important genes for both males and females. So a famous example of X-linked characteristics would be color blindness. So on the right-hand side of the screen here, you can see two circles. And those of us that have full red-blue color vision, we can see that there's a 29 in the middle of this circle on the top, and that there's a 74 in the circle on the one on the bottom. Okay. Now, if you can't see that, that's okay. Color blindness is actually extremely common and doesn't really cause any detriment to anybody in the modern world. And in our ancestral past, it may have been detrimental because maybe you couldn't identify whether or not a certain food source was a good thing to eat or whether it was poisonous. You may not be able to interact with your environment in the same way, but in the modern world, having some red blind color blindness is not the end of the world. Okay, so those are the two those are the essentially what sex-linked traits are. So let's talk a little bit about how you would calculate this using Punnett squares. Okay, so let's draw it out on our whiteboard again. I'll go back here to our whiteboard. Okay. All right, so let's look at the possibilities first, actually. Let's do another. Or we look at the genotypes and the phenotypes here. So we'll put the genotypes on this side, and then we'll put phenotypes over here. Oops. Let's do it like this, actually. All right. So for females, there's actually going to be 3 possibilities. And with these, you have to put the letter on the X chromosome. So you're going to write that both X's, because both females all have 2 X chromosomes, but you're going to put the letters right next to the X chromosomes. So a female could have 2 dominant alleles on both of her X chromosomes. She could have a dominant allele on one X chromosome and a recessive on the other, like this. Or her third possibility, she could have a recessive allele on both of her X chromosomes. So it would look like this. So if that female has a dominant C on both of her X chromosomes, then that means that she has completely normal color vision. We'll just put normal vision for short. If she has a big C on one of her X chromosomes, but a little c on the other, that means that she is a carrier for color blindness. We'll just put carrier. All right, and the only way a female ends up being colorblind, she would need to have the 2 recessive alleles on both of her X chromosomes. So that's a colorblind female there. Okay, now for males, since they only have 1 X chromosome, they're only going to have 2 possibilities. Remember, males have an X and they have a Y. So if that male has an uppercase dominant C on his X chromosome, and we don't put anything on the Y, because remember, this is an X-linked trait. So we put the gene on the chromosome that exists on. So we only have 1 X chromosome in males. So that's where we're putting the letter on or next to the X chromosome. So this would be a normal vision male, because he has the dominant big C on his X chromosome. And then the only other possibility for males, if that male has a little c on his X chromosome, then that means he's colorblind. Okay, so colorblindness is more common in males simply because they only have 1 X chromosome. So that means if their 1 X chromosome is affected, then they will have that condition. They will have colorblindness. Okay, so for this scenario, we have 5 possible genotypes and another 5 possible phenotypes. So for females, they have 3 possibilities. If they have a big dominant C on both of their X chromosomes, and they have completely normal vision. If they have the combo, if they have a dominant gene on 1 X chromosome and a recessive on the other, then they themselves have normal vision, but they are carriers, which means that their sons and their grandsons may have color blindness. And I'll show you how here in a moment. The only way a female is colorblind, she would need a lowercase recessive C on both of her X chromosomes. That would be a colorblind female. And then for males, there's only 2 possibilities. Since there's only 1 X chromosome, there's either a dominant allele on the X chromosome or there's a recessive. So males are either normal vision or colorblind. They cannot be carriers. All right, you guys with me so far on the possibilities?
Yes.
Perfect.
Yes.
So those are the possibilities for males and females when we're talking about an X-linked trait. So that could be color blindness, it could be hemophilia, anything that's X-linked, that's how you would essentially write out the possibilities. So now that we understand the possibilities, we can set up a Punnett square in a similar way. So let's say, all right, you guys told me in practice problem number 1 under sex-linked, what are the parents on check-in number 1? Let me go check here. Usually I have this memorized, but let me go check here. Okay, so a female that is a carrier for color blindness is crossed with a normal vision male. What percentage of the male offspring would be colorblind? All right, so we've got a female that is colorblind. Oh, sorry, a female that's a carrier, excuse me, a female that is a carrier. So to represent a female that's a carrier, so we're going to need one of her X's with a big C, with a dominant big C, and then we need her other X to have the recessive little c. Okay, so that's a female that's a carrier. So we're still setting it up in the same fashion. One parent's along the top, one parent's along the side. So in this case, we've got mom along the top, and then we're going to put dad along the side. So if dad has normal color vision, then that means he needs an X with a big C. And of course, he also needs a Y since he's a male. So then we need a Y here. Okay, so once we have it set up, it's the same as any other Punnett square. So here in this first box, we've got an X big C, X big C. Here in the second box, and always the dominant trait first, so X big C, and then an X little c like that. In our third box, we've got an X big C and a Y. Okay, and then in our last box, we've got an X little c and a Y. Okay, now here's where it actually gets a little bit trickier, because what it's asking is what percentage of the male versus female children will be colorblind. So if we're asking just about the female children, for example, we have to pretend that this part of the Punnett square doesn't exist because we're just looking at the female children. So every single time this mom and this dad have a female child, do they have any chance of having a female that's colorblind? Or either one of those females colorblind?
No.
No. So if it's asking about just the female children, this mom and this dad have a 0% chance of having a colorblind daughter. Now they do have a 50% chance of having a daughter that's a carrier for colorblindness, but they have a 0% chance of having a daughter that's colorblind. Okay, so we would write 0% of female children would be colorblind. Okay. All right, now what about the males? So for the males, we have to basically pretend the top half of the Punnett square doesn't exist because we're just looking at the males right now. We're just looking at the sons. So out of the sons, what percentage of these parents' sons would be colorblind? What's 1 divided by 2?
50%.
50%, because we're just looking at the son. So every single time this mom and this dad have a son or a male child, they have a 50% chance that that son will be colorblind. So we're going to write 50% of male children will be colorblind. Okay. All right, so we got 0% of the female children would be colorblind, but 50% of male children would be colorblind. Are you guys seeing why? Because this is usually where students get tricked up, is because it's looking at just the males or just the females. Just making sense? Hopefully. Yes. Okay. All right, so what we can look at next is our second one. Let's erase these here. All right, sorry guys, I'm not sure why the whiteboard jumps around so much. Let me just make another square, I guess. Okay. All right, so our second sex-linked Punnett square Let me just double check our scenario. So our scenario in our second sex-linked Punnett square example, a female that is colorblind is crossed with a normal vision male. What percentage of the male offspring will be colorblind? Okay, so we're looking at a colorblind female crossed with a normal male, normal vision male. All right, so we go here to our whiteboard. All right, so if it's a colorblind female, she needs, the only way a female can be colorblind, she must have the recessive little c on both of her X chromosomes. So we'd represent it as X little c, X little c. And then if the male has completely normal color vision, then he only has one X chromosome. So that means on his one X chromosome, he must have the big C on his one X chromosome. So he must be X big C. And then of course he's a male, so he also needs a Y chromosome here. Okay, so we set it up just like any other Punnett square, one parent along the top, one parent along the side. And then we fill in the little boxes with the dominant allele first. So we've got an X big C and then an X little c here. And then here in this box, same deal. We've got an X big C, X little c. And then in here in this box, we've got an X little c and a Y. And then in our last box, we've got another X little c and a Y. Okay, now same thing. It's asking about just the female children versus just the male children. So let's do the females again first. So in order to be a female, we need two X chromosomes. So we're just looking at the top half of the Punnett square. So out of the female children, would either one of those female children be colorblind?
No.
No. Okay, would any of them be carriers?
Yes.
Yes, 100% of the females are carriers. So we would write 0% of the female children would be colorblind. Oops, not sure why that disappeared. All right, 0% of female children would be colorblind. That 100% would be carriers. Okay, but the male children, so now we're just looking at the bottom half of our Punnett square. So out of the male children, what percentage would be colorblind of the male children?
100%.
100%. Excellent. So 2 out of 2, 100% of male children would be colorblind. Okay. All right, so those are our likelihoods. So every single time this mom and this dad have a child, anytime they have a daughter, they know for a fact that their daughter won't be colorblind, but she will be a carrier for colorblindness. And every single time this mom and this dad produce a son, we know that 100% of their sons, every single time they produce a male child, that male child will have colorblindness. Is this making sense?
So basically a female can carry it, but she's not necessarily colorblind, meaning she can pass it on to her children as well.
Correct. Yeah. So if a female is a carrier, that means that herself, so like these daughters here. So these daughters, they have completely normal color vision for themselves, but that means that they may actually, because they're carriers, and may be more likely that they're going to pass that along to their children, especially their sons. Okay, now the only way a female would end up being colorblind, the only possibility would be if her mother was a carrier and her father was colorblind. Okay, that would be the only way, or of course, if they're both colorblind, but the only kind of sneaky way, let's say, that a female would be colorblind would be if she has a mother that's a carrier and then a father that's also colorblind like this. Okay, because we're inheriting one gene, one homologous chromosome from mom and one homologous chromosome from dad. So here in this little box, in this scenario, here, let me erase these just so it won't confuse anybody here. All right, so in this first little box, we've got an X big C and an X little c like that. And then here in this box, we've got an X little c right there. And then here in this box, we've got an X big C and a Y. And then here in this box, we've got an X little c and a Y. All right, so in this scenario, looking just at the female children, so what percentage of the female children would be colorblind in this scenario? So we're looking just at the children that have two X chromosomes, so just the top half of the Punnett square.
50%.
50%, excellent. Remember, the only way a female can be colorblind, she must have a recessive little c on both of her X chromosomes. So in this scenario, 50% of female children will be colorblind. Okay, now the other 50%, so looking at the other female right there, she is not colorblind herself because she has at least one dominant big C, so she is not colorblind. She has normal vision herself, but she is a carrier for it. So in this scenario, 50% of the female children will be carriers. Okay, now what about the male? So now we're just looking at the bottom half of the Punnett square. What percentage of the males would be colorblind in this scenario?
50%.
50%. So just this one right here, this male would be colorblind. This male would have normal color vision. So in this scenario, 50% of male children will be colorblind. But in general, if you're not talking about gender or biological sex, I should say, these two parents have a 50% chance, whether or not the child is male or female, they have a 50% chance of having a child that's colorblind. They have a 25% chance of having a child that's a carrier for colorblindness. And they have a 25% chance of having a child that has normal vision. So for these two parents, they have a far greater likelihood of having a child that's either colorblind or at least a carrier for colorblindness. Because mom's a carrier and dad's colorblind himself. Other questions? And so somebody raised their hand. I'm sorry, guys, when I'm here on these different screens, sometimes I can't really see the chat box. So, and raising your hand is fine. Just, you know, also if you can ask the question too, I can't usually see the chat box when I'm in these different screens. And unfortunately, I don't have a TA to assist me with the chat box. I wish I did. No questions? Okay, we're not going to do breakout rooms today just because we only have about 10 minutes left. But let's do one more example of something. Do you guys want to do one more basic Punnett square? Do you want to do one more AVO blood group? Do you want to do one more sex linked? What do you think?
Can we do
Let's
an
do another
AVO?
sex.
Yeah, can you do one more sex link?
Sure. Yeah, let's do one more sex link. So let's say this time we're talking about hemophilia. So hemophilia is a blood disorder that has the same exact method of inheritance as color blindness. It's another X-linked trait. So for males, they're more likely to have hemophilia because they only have one X chromosome. So for females, there are possibilities. They could, and we're going to use H's this time. So if they have an uppercase H on both of their X chromosomes, they do not have hemophilia. So we'll just put normal blood for short. They can be carriers. So if they have one normal uppercase H on one X chromosome, a recessive lowercase on the other, then they are carriers for hemophilia. Now, the only way a female has hemophilia, she would need to have a recessive lowercase h on both of her X chromosomes. So that's a female with hemophilia. All right, so for males, so just like with color blindness, for males, they're only going to have two possibilities because they only have one X chromosome. So if they have an uppercase H on one of their X's, on their one X, I should say, if it's X, H, big HY, then that's a normal blood female, normal blood male. So the only other possibility would be a little lowercase h on the one X chromosome. So an X lowercase HY, that would be a male with hemophilia. So it's the same pattern of inheritance as the color blindness example. We're just using H's now because we're talking about a different trait. We're talking about hemophilia instead of color blindness. So let's say that mom is a carrier. So if she's a carrier, that means that she has a big H on her one X, and then on the other X, she's got a lowercase h. So mom is a carrier for hemophilia. And let's say that dad is normal. He has normal blood. So that means on his X chromosome, he's got an uppercase H representing the dominant allele, and then he's got a Y. All right, so here in the first box, you guys tell me, what would I put here in the first box?
XH, capital H.
And what kind of H's, big or little?
Big for both.
Excellent. So on both X's, there would be a big uppercase H. Okay, well, we'll go here in the second box.
XH.
Okay, and what kind of, an uppercase and a lowercase? Perfect. So in the first X, we've got an uppercase. Second X, we've got a lowercase like that. So we've got an X big H, X little h. All right, what goes here in the third box?
XHY.
And what kind of H?
Capital H.
Excellent. So we've got an X, capital H, and a Y. All right, and what goes in the last box?
X lowercase h y.
Beautiful. Last box is an X, lowercase h, and then a Y. Okay, so we've got it all set up now. So generally, the sex-linked Punnett squares are going to ask about just the male children or just the female children. So let's look at the female children first. So we've got one female child here. So she's an X big H, and we've got another female child here. She's an X big H, X little h. So out of the female children, what percentage would be hemophiliacs or have hemophilia?
50%.
Excellent. So out of the female children, 50% would have hemophilia. Or I'm sorry, excuse me. Sorry, so 50% would be carriers. Sorry, guys, 50% would be carriers. And then the other 50% would be normal blood. Okay, so in order to have hemophilia, a female would have to have two of the lowercase h's. The only way a female has hemophilia, she needs two lowercase h's, right? So that's not the case for either one of these. There's one female that has an uppercase h and a lowercase, but she would not have hemophilia because she doesn't have two lowercase h's. So out of the females, 50% would be carriers and 50% would be normal blood. Okay, now what about the males though? Out of the males, so now we're just looking at the XY. So we're looking at this one and we're looking at this one. So out of the males, what percentage would have hemophilia?
50%.
Excellent. So out of the male children, we have 50% hemophilia, and then we have 50% normal blood. Okay, so out of this scenario, it is not possible that these two parents could produce a child, or a female child, I should say. They cannot produce a female child that has hemophilia. They have a 50% chance of producing a female child that's a carrier, and they have a 50% chance. So there's the carrier child here. That's the carrier. So an X big H, X little h, that's a carrier female. And then they have a 50% chance of having a child, a female child with normal blood. As far as the male children go, it's even. It's 50-50. So out of the male children, they have a 50% chance of producing a male child with hemophilia, and they have a 50% chance of producing a male child with normal blood. Okay? Is that making sense?
Okay.
Is anybody still lost? Everybody's good? Okay, so let me go to share screen here so I can show you what to do for homework. So the good news is, those of you that were here today and were taking notes and paying attention, you guys are actually done with your homework. All you have to do is then upload it. Well, it's in quiz format, so all you have to do is then fill in the answers. But what I went over with you guys today is actually identical to what your quiz is. Okay, so we essentially did check-in assignment number 1 together in class today. So those of you that were here and those of you that were paying attention and taking notes, you're actually done with your homework. All you have to do is upload it here on Canvas. So you go here to check-in assignment number 1, Punnett squares. And then here, I'm going to show you guys in student view so you can see what you're going to see in student view. So it says that this task should take about an hour of your time, but you guys have already spent that hour. You already spent the hour here in class. And it's going to, if anybody wants to watch this again, I do actually have a recorded video of me going over it. I'm using a Microsoft Doc here. But there's a recording if anybody wants to watch it again. And there's some, YouTube clips that I found helpful for this topic. And then you just go down here and you go to take quiz. Okay, so essentially when you go to take quiz, all you're going to do is fill in the answers. So you've got a couple vocab words that we went over in the PowerPoint slides. So you're describing some of the vocab words. And then once you get down past the vocab words, then these are the Punnett squares. And these are actually identical to the Punnett squares that we did here in class. So for those of you that were paying attention and took notes, all you have to do is then input your answers here in quiz format, and then you're done with your homework for this week. Assuming that you've already done your reading, of course. So at this point, you should have read through chapter 3. Okay, you should have read through chapter 3 in the Essentials textbook. And next week, we're going to be delving into chapter